How many of you have come into science after watching science fiction when you're this much or this much? The major of science researchers are influenced by science fiction, and it plays a great role on our imagination. And I grew up like that. And when I say science fiction, how many of you remember spaceships, Star Wars, intergalactic spaceships traveling from one planet to another planet and everything, right? And let me say, how many of you imagine talking to bacteria, talking to viruses? Let's make friends with viruses, they're only up there. Or how many of you imagine walking on a cell membrane of a bacteria or fixing the genes with your hands? This is not what we imagine, because most of our science fiction has been driven by what we see outside than what's inside the life. Life at a molecular level is highly complicated to imagine. In my hand, I have 3D models of few proteins which I designed, and they could be a potential cure for cancer. And I made them on literally computers using interdisciplinary skills here. So these computers are the same ones which 60% of people on the earth have there in their households. To me, these shapes, I call them beautiful. But if I ask someone in the audience, I'm sure you would say they're just spheres or globules, or they're just dirty looking colors, one for men, one for women, or vice versa, whatever. So, when we have these molecules, the structure at an elementary fundamental level in biology is very important, and it's not what it looks like. For example, this protein, what looks like a globule, is made up of helices, sheets, and coils. Can we imagine that by looking at this? And this is the complexity people were dealing with when they're dealing with biological structures. In 1953, Rosalind Franklin was working on solving a structure of a biomolecule. And she had similar problems, and she was using X-ray diffraction. When I say X-ray diffraction, it might sound complicated, but if you ask someone to turn on the light, you go and turn on the light. But if you ask the same question in a science laboratory, they would say, what light? Light has many types, and X-ray is one type of that light. And when you pass that light through some pores, you get a pattern called a diffraction pattern, which might look like this. And these patterns depend upon the structure of the molecule. Now, at the end of her discovery, she was on the Nobel laureates list. She did not win because she'd passed away of cancer, but she was on that list, and she discovered this. This today we know as the structure of the double helix DNA. And this structure is responsible for all the miracles what we're observing today in cancer, in therapeutics, and whatnot. This is DNA. Thank you.